Okay, test. Okay, testing my mic. Okay, so mic is okay, camera okay. Okay, so now is already 8 o'clock. Okay, so we will start our class now. Okay, so last week we already finished up the chapter 10, reproduction, development and growth. So today and tomorrow we will finish up the chapter 11 homeostasis. So in this chapter there are 4 subtopics that you need to learn. Importance of homeostasis, liver, and osmoregulation, and final one is osmoregulation in plant. Okay, so today we will only proceed to 2 topic only: importance of homeostasis and also liver. Okay, so first of all, you need to know what is the meaning of homeostasis. Okay, so as we know that homeostasis is the regulation and maintenance of your internal environment in your body organism, meaning that it is the automatically control process to maintain that your internal environment of your body can be at the best survival to you. So meaning that the human, any organism will stay at the best environment that can fit based on the homeostasis mechanism. So if let's say there are the outer environment, maybe outer environment that is the temperature, because you will see that why if you are at the temperature like negative, or only in winter country only 1 degree Celsius of 10 degrees Celsius, but humans still can survive, right? But if you see at desert up to 50 degrees Celsius, but still people at there still can survive. Why? Because of this homeostasis. This homeostasis can help the organism to maintain the constant internal environment. Because it need to maintain your internal environment is always the same. It will not like change frequently, because it need to maintain the metabolism rate and also enzymatic activity inside yourself. Okay, so if you see the basic principle of the homeostasis, so actually this is similar what you see in about the nervous system Actually homeostasis is controlled by nervous system too. So in general you need to have a stimulus first. You need to have a changing first, any change. So in human, I think in previous chapter actually you already learned about the homeostasis actually, like the chapter 7, breathing mechanism. Breathing mechanism, what the change? The stimulus is when the carbon dioxide concentration in your blood are changed. Either it decrease or increase, right? But normally it's increase, right? It's increase, right? So if your carbon dioxide concentration increase, what are the effects? What are the effector? Effector is your breathing rate, your ventilation rate will be increased, right? This is for breathing mechanism. And in chapter 10 about the hormone positive feedback mechanism, right? Positive feedback mechanism about the oxytocin during the patient during the birth rate, it increase the concentration of your oxytocin, right? And then the increasing concentration of oxytocin will also stimulate the uterine wall, muscle wall contraction. This is our effector, and this effector will provide the response to stimulate again the stimulus to secrete more amount of oxytocin. So it is the general principle. So the general principle of homeostasis is you need to have stimulus first. Any change, like your blood pressure, your breathing rate, your carbon dioxide concentration, your blood glucose level, any stimulus change inside your internal environment, when they are the changing detected, so your body only can maintain the internal constant environment only, right? So like I give an example like blood pressure. Normal human blood pressure is only 130 over 80 only. If your blood pressure is too high or too low, it's unlogical. It is the abnormal situation lah. So in basically your homeostasis mechanism, when the blood pressure is changing, so it will send the stimulus to the receptor. Normally this sensory receptor. So normally this receptor normally is located in the brain lah. Also when the receptor is stimulated, it will send this signal to the control center. So normally this in your brain, it change to the control center. So you need to give the instruction that the muscle, give the instruction to the effector, normally is muscle or your endocrine gland, to ensure that your internal environment is changed to the set point, so that the blood pressure, your set point is 120 over 80. So when your blood pressure is too high, so your control center will send the signal to your muscle, to your heart muscle, or to the blood vessel to slow down, maybe slow down the heartbeat, to decrease down your blood pressure. Or if your blood pressure is too low, so you want to return the set point, so you need to increase your blood pressure. So this one control center, it will modify the signal the signal received from the receptor, and then send this signal to effector, so that the effector can function based on the signal from the control center. Either muscle either you want to relax or contract, and then your gland either you want to secrete the hormone or they don't want to secrete hormone. And then once they already receive the stimulus from the control center, then it will respond lah. So for muscle, either you want to contract or relax, or for the gland, either you want to secrete the A hormone or another hormone, or directly no hormone as well, or you want to increase the secretion of hormones. So this is the response function. So when the response function is function, so it will check the value, the internal environment will become maintain constant. So your blood pressure now will maintain to the set point, which is 120 over 80 mmHg. Okay, so it will return back. So when the stimulus is coming again, so the homeostasis will be repeat again. So this is the basic principle of the homeostasis. So before you enter this chapter, actually in chapter 7 and also chapter 10, you already learn example, but I think in SPM you also already learn too. Okay, so in this chapter, these principles are very important because you will see it this many lah. Okay, so as you guys know, our mechanism in homeostasis, there are two main class of mechanism: positive feedback and also negative feedback, right? So I think everyone should learn about the negative feedback. So positive feedback, one of the example is oxytocin during the birth. So I already explained last week about the last chapter. During the pituation, during the birth process, or positive feedback mechanism will be occur, meaning that oxytocin will increase the contraction of uterus, and then this contraction of uterus will increase more and more oxytocin be secreted. So this is one of the example of positive feedback. So what is the meaning of positive feedback? Is to amplify the stimulus. Rather than to reduce the stimulus, positive feedback need to amplify. Dia kena membesarkan stimulus itu. Okay, but in human, normally positive feedback mechanism, one of the famous example is human birth only. And then most of the mechanism is negative feedback. So negative feedback meaning it reduce the initial stimulus. You need to change the stimulus into the return into the set point. Like blood pressure, if your blood pressure is too high, then you want to your bodies want to decrease your blood pressure. But if your blood pressure is too low, then you need to increase your blood pressure until it return to set point. And also your carbon dioxide concentration, right? So if your carbon dioxide concentration is too high, or hydrogen ion is too high, so you need to reduce the carbon dioxide concentration in your blood. So how it do? It will increase the ventilation rate, right? Okay. So this is a negative feedback mechanism. So there are many examples. These three are in your syllabus that will learn in this chapter: thermoregulation, osmoregulation, and also blood glucose level. Actually, there are more. Another one is blood pressure, and then you already learn breathing mechanism. These two also are the example too. Okay. Okay. Now I go to importance. So why the homeostasis is very important. So actually before that I already mentioned something. So one of the important is it interplay between external factor and also internal factor control mechanism. So it is like a control or intermediate between to control the external factor and also internal factor. So this equilibrium will produce the steady state, meaning that your internal environment is relatively constant even though your external environment was changing significantly. So in your internal environment, like the body temperature, one of the example, body temperature, your internal environment is normally 36 degree until 37 degree only, right? This is your internal environment, and then your outer environment maybe will change. Maybe like now it's hot sunny day, right? Hot sunny day. So maybe your external environment can up to 40 degrees Celsius, but you still can survive. And your internal body temperature still is maintaining at 37 or 36 degrees Celsius only. So this is the importance of the homeostasis, because it produces the steady state. Whatever your external environment change, your internal environment will be maintained constant. Okay. Another one is your physiological activity by the response to stimuli are not instantaneous, meaning that your internal mechanism will not change the environment immediately. So meaning that when there are the response to the stimulus, it is the instantaneous activity, meaning that your internal mechanism, when you want to change your internal mechanism, actually will not change the environment one, and then this process is not immediately processed. It not like when your outer environment change significantly, then it will starting to stimulate. No, it need to take time lah. Okay. As a result, homeostasis moderate the change of internal environment only, not elimination internal environment. So it is not eliminating the internal environment, but change the internal environment. And then homeostasis is also enhanced by adaptation in organisms like hormone and effector in the body to reduce fluctuation. And also it is a biological system to function efficiently, so there is less energy wastage. Also to allow organism can live in wider range of habitat. So if no homeostasis was there, meaning that the human only can survive at tropical forest area only, right? And then you cannot survive at winter country, and then you also cannot survive at very hot country. So there are no such thing happen. So why? Because of homeostasis. So homeostasis let human can survive in this wider range of the environment, and also allow enzymatic reaction and cellular activity go normal in the body. Okay, so this is all about the importance of homeostasis that you need to know lah. Okay, now we go to the one of the example of negative feedback mechanism in homeostasis, which is thermoregulation. So this is about the maintain your body temperature within a normal range. So there are only two conditions. Either it is very hot, you are at very hot condition like at 50 degrees Celsius, or you are at very cold condition, meaning that you are at the winter country like maybe 10 degrees Celsius, or even in negative. negative degrees Celsius like that. Okay. So actually in the world, in the animal, there are two main type of organism lah, and either you are endothermic or ectothermic. So in our syllabus, we will only learn about endothermic only. Ectothermic we not learn. In our syllabus we only learn about endothermic. So endothermic like human. So what is the difference between these two? So endothermic actually is the organism are usually warmed by heat generated by metabolism of the cell. So meaning that endothermic organism it will produce the heat itself. It will not receive the heat. So meaning that this organism, they will maintain their internal body temperature as well. Like human, our internal temperature is only 37 degrees Celsius, then it will maintain as well. Maintain along the time. Or wherever you go to which kind of country, you go to summer country, if up to 50 degrees Celsius, or you go to winter country like North America, or you go to Antarctic, you go to the North Pole or South Pole, even though you go there, but your internal body temperature still maintain the same. Compared to ectothermic is this kind of organism is gain the heat from the external source, meaning that this organism, the cells, will not produce the heat one. It will not produce the heat. It will just take the heat from the external. So this kind of organism, the body temperature will change based on the environment. So when you place in the cold country, then the body temperature will become cold. When you place this organism at the hot sunny day, like 40 degrees Celsius, then internal body temperature also is becoming 40 degrees Celsius. It will not like the endothermic. Endothermic will maintain all the time of your body temperature, but ectothermic will not maintain. It will change according to the environment. When the environment is hot, then body temperature is also hot. When the environment is cold, then your body temperature is also cold. So you need to alert lah this. Though in our syllabus, we will only learn about thermoregulation in endothermic only, so including human lah. Okay. So in our syllabus, you need to learn about the thermoregulation in endothermic occur these six mechanism: insulation, blood vessel contract relax, counter current exchange, evaporation, thermogenesis, and also behavioral response. So this is all the response, all the physiological and also behavioral response when the endothermic organism face either hot day or hot temperature or cold temperature, and you need to regulate your body temperature. Okay, so we see it one by one. Okay, so insulation is only occur during the hot condition. So in hot condition. So insulation reduces the flow of heat between the animal body and its environment. Insulation is only respond when the organism is at the cold temperature, meaning that you are at the cold temperature environment. So it need to reduce the flow of heat between an animal body and its environment. So you need to reduce the heat transfer from the body temperature for the internal body to the environment, because you are already very cold. You are at the very cold environment. So if your heat is transfer from the inside to outside, then you will die. Because your body temperature will decrease. So insulation will play the role to reduce your heat between the animal body and also environment. So this one is only occur during the cold day. When you go to the aircon room, 60 degrees Celsius like that lah, or you go to winter country like that. Okay, so normally insulation is found at the both at the body surface and also beneath layer. So this normally is found at the skin lah. So either it is body surface like your hair, the animal hair, and also the beneath layer, the skin, the inner layer of the skin. So normally it's adipose tissue. So this one why we say that the people which is more fat, the fat people will be not more struggle and can more tahan at the cold environment. So awak boleh tengok punya orang yang gemuk tu memang dia tak boleh tahan panas punya. Memang dia tak boleh tahan panas, dia sangat nak aircon punya. Kerana dia ada ini adipose layer, which is fat lah. Okay. So normally in the insulation, so insulation process will secrete the oily substance that repel water, protecting your insulating capacity of your feather or fur. So normally this one is more compared to the birds organism, and then the animal can adjust their insulating layer to regulate your body temperature lah. Okay, so during cold environment, organism, your fur and feather are fake. Will be rise. So you will see your hand. So I think some of you your hand have fur and feather, right? So when you are at the cold environment, your fur and the feather will be rise out, right? Will be at rising state. So this action trap. Why you want to rise your feather and your fur? Because you need to trap the thicker layer of air, so increasing your effectiveness of your insulation. And then the organism lack of fur like human must really primarily fat for insulation. So compared to other organisms like dog, like cat, they have fur, more fur and feather, right? But in human, we are lack of fur and feather compared to this animal lah. But some of the human, they containing more fur and feather compared to other human. Okay, but never mind. But in general, human is lack of fur compared to the animal. So normally human is rely on your fat, if which our adipose tissue lah. Okay. But in mammal organism, the swimming process, they will swim in water colder than their body cold. So this species normally will spend at least part of the year nearing freezing policy to transfer your heat to water. Heat to water occur more rapidly than heat transfer to air. Okay, so survival under this condition is made possible by an evolutionary adaptation called blubber. A very thick layer of insulating fat just under the skin. So yeah, this is just want to ask. I want to give you that due to insulation process, due to you are at very cold environment, so you need to increase your fat amount. So that is why that you will see that United States country, especially got the winter day country, their eating habitat will totally differ from us. Their eating habitat, normally the quantity of the food is very large amount, very in very large amount. Normally in Malaysia, especially girl, you cannot eat finish one. So why? Because they need to increase the heat production. So they need to increase the rate of insulation. So they need to increase the amount of fat. But this is very unhealthy. But there are no choice because they are survive at that kind of temperature, that kind of environment. So they need to do this. Okay, so they can maintain the body core temperature without require much more energy from food than land mammals of similar size. Okay, so that's all for insulation. So next I go to blood vessel contract and relax. Okay, so for this one, there are two conditions. Either your blood vessel is contract or blood vessel is relaxed. Right. So either your blood vessel is contract and blood vessel is relaxed. So when your blood vessel will be contract, Okay, so this one is to maintain your blood flow to either want to increase the heat transfer from your blood from the blood to the outside, or reduce the heat transfer of your blood to outside. So it is a circulatory system provide a major role of heat flow between the interior and also exterior. Okay, so in terms of changing, so many animals will alter the amount of blood flowing between their body core and also their skin. So the most important thing is during the hot condition, your blood vessel will be relaxed, or we known as vasodilation. I recommend you guys using this term vasodilation, meaning that your blood vessel will be dilated and increase your blood diameter. So why you want to like that? Because you are very hot. You are very hot now. You imagine you are in the very hot condition, so that in your body core temperature also actually should be increased temperature. Your body core temperature should be increased, so that they need to release out all this heat. They cannot be too high one. They cannot be too high one. You need to maintain your body temperature at the 36 degrees Celsius. If your body temperature is up to 38, 39, so they need to release out this heat. So how to release? So normally they will release through the blood, so that all the blood need to increase its diameter, so that more blood can be flow throughout the skin surface. So many blood flow, the rate of blood flow to the skin surface will be increased, so that heat transfer from the blood vessel to the exterior body will be increased, so that more heat will be transferred, more heat will be loosed out lah. Okay. Another condition is during cold condition. So when you are very cold, so this is a reversible, so meaning that your blood vessel will be vasoconstriction, meaning it will contract. You need to reduce your blood flow to the skin surface. Why? Because when you are in cold condition, meaning that you need to maintain your high body core temperature outside the environment. So maybe you are in 10 degrees Celsius only. So you need to maintain the high temperature 36 degrees Celsius, so you cannot let your blood many blood flow through to the skin surface to increase the heat transfer between the interior and exterior of the body, to lose out all the temperature. Cannot, because you need to maintain. So for now your vasoconstriction, so your blood vessel will be contract, so to reduce the blood flow, so that it can maintain your heat inside your body. So this is the blood vessel contract and relax. Okay, next is about counter current exchange. So this one is more special to the cold condition environment. Hot condition will not follow this. It will only more special to the echo environment only. So how this open? Okay, so this one is the blood. It is similar to the what I say before, blood vessel contract and relax. But this one compared to before, ni blood vessel and contract relax, it is the heat flow between the interior and exterior, right? But this one is heat flow between the artery and vein. So in general, your artery will carry the warm blood. Artery will carry the warm blood compared to vein. Your temperature of your blood in artery will higher than your vein one. So into counter current exchange, meaning that the heat inside the heat from the artery actually will be transferred to the vein. So when the blood vessel, especially the blood vessel in the artery and vein, are close contact to each other, the heat exchange will be occur. So heat exchange will be occur. Why? Because artery and vein are flow in opposite direction. We know that artery will flow from heart into all part of body, right? So normally it's downward, but vein will be transfer your blood from the blood any tissue layer to the blood, right? So it is upward. So normally in general, your blood flow of your artery and vein are opposite to each other. So this arrangement will facilitate the heat transfer from arteries to vein along the entire length of your blood vessel. So especially when the artery and the vein are near to each other, this heat transfer process will be occur rapidly. Okay, so near the end of the leg where artery blood has been cooled to far below the animal core temperature, your artery will transfer heat to even cooler blood in an adjacent vein. So this one just want to tell you that when your artery blood, oxygenated blood is near to the blood leg already, so your arterial blood will be cooled down from all temperature, but although it is cooling down, but the temperature still are higher compared to vein, so the heat exchanger will still can occur. So at number 3, as the blood in the vein approach the center of the body, it is almost as warm as the body core, so minimizing the heat loss that result from supplying blood to body part immersed in cold water. So this one is to actually also want to reduce the heat loss, so that not only you have a vasoconstriction, because now your vein temperature, your blood vessel in the vein now is also quite high, also similar to core temperature, so that heat exchange between the interior and exterior body at the skin surface will be occur at very condition, very situation. Okay, so this is the explanation as well, so you can read yourself lah. Okay, so normally I already explain almost all things lah. Okay, so just I want to highlight that in mammal organism, they often use this mechanism to keep the main swimming muscle warm, so that to enable vigorous activity. Okay. Another one is about evaporation. So this one is only occur during hot sunny day only. Evaporation, right? Evaporate meaning berpeluh, right? Okay, berpeluh. So you want to, if your environmental temperature is higher than your body temperature, so normally is in your hot condition, like 40 degrees Celsius like that, your evaporation can keep your body temperature from rising. So evaporation, as you learn in chapter 1, water topic, about the chemical properties of your physical properties of the water molecule, we know that one of the property is water have high heat of vaporization property, right? Meaning that you want to change the state of the water molecule require high amount of energy. So this one will probably to evaporate cooling effect as well, so that water molecule with the greatest kinetic energy will leave as gas, making the remaining liquid water cooler. So so this is the basic theory that you already learned in some one. Okay, so water will absorb considerably heat when it evaporates. So this heat will be carried away from the skin and then respiratory surface from the water vapor. Okay. So evaporation of your sweat from the skin dissipate body heat and help to prevent overheating on a hot day, so or when excess is generated in strenuous activity. So normally evaporation, it will contribute sweating process, so to prevent your overheating in your body lah. And also some birds have a punch richly supplied in blood vessel in the floor of the mouth, fluttering the punch increase evaporation. Okay, so this is the about evaporation. Next is about thermogenesis. Okay, so this process actually is also not similar. This process is also only occur during cold condition only. because you need to know that the thermogenesis is the increase your muscle activity for moving and also shivering. So normally this one is only occur at very cold condition. When you are in the very cold temperature environment, so thermogenesis maybe take place. So endotherm will have very heat production to make changing rate of the heat loss. So this is actually production of the heat, but it increase the production of heat when it is stimulated lah. Okay. So normally for small organisms like bird, shivering can make them reactive and hold their body temperature nearly constant in very low environmental temperature. Okay, so this is for bird. And then like the insect too, so shivering can shiver their wing to be wing before fly first. So that is why that you see that some of the insect they fly, they will not directly fly, they will have a ready first, they have the preparation first baru boleh terbang. Okay, so normally this one is chemical reaction and then cellular respiration and also accelerate your warm up, flight motor, and I believe fly even in cold air. And another one is endocrine signal released in response to cold cause mitochondria to increase their metabolic activity and produce heat instead of ATP by non-showering thermogenesis. So this is another process we call as non-showering thermogenesis, meaning that there are the endocrine will release your response to increase your metabolic activity. Okay. So activity. So this one is compared to this one is totally different. This one is because your muscle activity is already changed, so it will act as a stimulus. But for this one, because in mammalian, endocrine signal released in response to the cold actual mitochondria to increase their metabolic activity. So will produce instead of ATP by non-showering thermogenesis. So for this one is this patient will not have a showering symptom, just like a former patient, but then when they are very cold, they will totally pingsan. Okay. Some mammalia also have a tissue called bowel fat in their neck and also between their shoulder, so that is specialized for rapid heat production. Okay, the last one, behavioral response. So this one is more easy lah. So either when you are in hot condition or cold condition. So normally when you in cold condition, you need to automatically you will find the warm place first, right? And then you also orienting themselves toward heat sources and expanding their portion of the body structure exposed to the heat source. Another question is about when hot they bath move to cold area or turn in another direction, minimizing the absorption of heat from the sun. So normally when they are in very hot condition or when they are bathing or they are moved to the cool area, they will minimize the heat absorption from the sun. Okay. Just now, I just mentioned about the thermoregulation in general terms. Now we proceed more specifically in terms of human skin. So in human skin, as we know that in human skin, there are three main layers, right? Three main layers: epidermis, dermis, and also subcutaneous layer. So epidermis and also dermis, they are already containing many blood vessels, containing hair, and also containing lymphatic vessels, and also containing sweat gland, containing hair, hot receptor and cool receptor as well, right? All in the dermis. And also epidermis normally is containing the keratin, and also fur. So this is the overall structure of the human skin. Okay. Now we go to the negative feedback mechanism in thermoregulation in human. So as you guys know that when the human, you are facing very hot temperature like this, when the environment temperature is starting to increase, so that your body temperature also increase, also increase, so that it will send the stimulus to your brain. So this brain stimulus will provide the cooling effect to your muscle and also blood vessel. So for your blood vessel, because it's hot, right? So what your blood vessel will do? It will dilate, right? Will dilate and radiate from the skin surface. And also inside your sweat gland will also secrete the sweat and also provide the evaporation process. Okay, so this one is in hot horny day. So I just want to highlight the heat loss central. So this heat loss central is very important central that are located in the anterior hypothalamus places, activated by increase in hot blood temperature and also send the impulse to the effector for the response. So when your nerve signal will transfer the muscle of vessel, causing the muscle to relax. So this one will causing vasodilation lah. So vasodilation will dilate and widening your blood vessel lah. And then this will cause your vessel diameter increase and your more blood will be flow to the skin surface. So that is why that during hot sunny day, your face will be very red color. More red color one, because you need to increase the thermoregulation rate lah. Okay. The insulating layer also in human we also contain insulating layer, right? So insulating layer will decrease the rate of insulation. Okay, so I already mentioned insulation process is only occur when the temperature is decreased. It is decreased. You are at the whole temperature, but if you are in hot temperature, so insulation will decrease the rate lah. Rate. Also, the sweat gland also stimulated to secrete more sweat from the skin surface, and also evaporation from your skin surface occur to prevent the body core temperature. So this heat will carry away from the skin in form of water vapor. Okay, so behavior response also will take place to decrease your body temperature lah. So this one similar to before lah, to previously. So what are your response? When you are very hot, what will you do? You will bathing lah, starting to mandi kan, or awak akan pergi ke ada aircon punya area kan, or you were starting to drink the ice water right, drink the cold drinks like that. So okay, so this is during hot day. So now we proceed to cold day. So what you learn before in hot day, so this is a reversible reaction lah. So what I need to highlight is there are also another center we call as heat gain center, which is located at the posterior hypothalamus. Compared to hot day, the heat center that will be stimulated is heat loss center. You need to lose the heat, so we call as heat loss center, which is located at anterior hypothalamus. But in cold day, it is the heat gain center, because you need to receive the heat. You don't want the heat released out already. You don't want your heat loose out from your body. So it means you need to receive the heat, maintain your heat, gain your heat. Okay, so this heat gain center is located in the posterior hypothalamus. The different one is anterior hypothalamus, one is posterior hypothalamus. So you need to differentiate them. Okay. So normally this center will transfer to the effector and then respond to trigger all the response that I mentioned just now. So one of the mention is your contraction vasoconstriction. Your blood vessel will contract, and then your insulation layer will increase the rate of insulation, and then you will pull up your hair okay. And then another one is your increase the thermogenesis rate to increase the heat production. And also in human normally we have the non-shivering thermogenesis, meaning that endocrine will send the signal to cause your mitochondria to increase their metabolic activity to increase large amount of heat and also ATP. Okay, and also human consists of the tissue called bile fat that is surrounding your neck and also between the shoulder. Okay, so this tissue actually can generate heat lah. Okay. Okay, not only that, we also containing the hormone. Your adrenal gland will stimulate your adrenaline, and then also stimulate the thyroid hormone, and also stimulate the ACTH hormone to increase your metabolic rate. And then this behavioral response, of course, if you are in a cold day, what you will do? You will wear more clothes, wear more shirt, wear jacket, and then also take place to increase the body temperature to that set point. So you will sit at the warm places, right? You will stay at the aircon room, 16 degrees Celsius, too few hours. When you are very cold, you will go outside, right? Outside more warm. And then you will orienting themselves toward heat sources. You will find any heat sources. You will go to chivalry, like that, right? Like that. Shake your hand, or you will movement, you will walk around, kan jalan-jalan sini, jalan sana, to increase your metabolic rate activity. Okay, so that's all for about the importance of homeostasis topic. So in this topic, you only learn about definition of homeostasis, importance of homeostasis, and also thermoregulation in endotherm. So far, any question? Okay. Okay, so I already finished the first subtopic lah. So now I will enter the second subtopic, which is liver, which is totally new to you that you never learned before in SPM, except the negative feedback mechanism of the glucose level. Okay, just now all the topic about the importance of homeostasis, definition of homeostasis, and also thermoregulation you already learned in SPM, right? I think you almost learned in SPM, so maybe you are very familiar with it. But in liver, although in SPM you have mentioned about liver, but I think you not learn about the detail. So in this topic, this is very important topic. Why? Because this topic coming out many times in essay question, especially the metabolism of carbohydrate and also metabolism of protein, coming out many years in past year. I can say it already coming. I think this along this 11 year from 2013 until 2023, 11 years including repeat, already coming out 6 to 7 times already. I think last year your last batch, your senior 2023 ulangan, it coming out again. So this topic is very important. You need to very focus lah. Okay. So first you need to know about the liver structure. Okay, so we know that liver we are separating into two loop side. Either you are separating into right loops and left loops and also right loops. And then inside the liver, when you enlarge your liver, you will see actually liver is arranged in lobule. We call as lobule structure. You can say or in other terms you can say is in hexagon structure one. They are arranged dekat hexagon punya. Polygon, hexagonal shape. And then inside each lobule, you contain the one lobule like this. It will arrange like this. This is the single lobule that I enlarge. If you enlarge, and then if you enlarge again, you will see this kind of structure. So this is actually the basic unit. Any part lah. Awak boleh tengok ada banyak part kan, ada banyak sini kan. Kami hanya enlarge satu unit je dari sini. So you will see here, this is the basic cross section of your liver lobule that you need to know the liver structure. Ini memang awak kena tahu. In exam, normally in structure question, maybe coming out in structure question, it will coming out this kind of diagram and then label lah. What is A? What is this? What is this? What is this? Or in objective question also maybe will come out objective and structure lah normally. But in essay I think will not come out. Essay will come out in liver function. Okay, so you need to know that in liver we containing 3 blood vessels, 3 types of blood vessels, very special. Compared to human, compared to other part, in general, human will only containing 2 vessels, right? Either you are artery or vein only, right? Artery carry oxygenated blood, vein carry deoxygenated blood. But this liver very special. It containing 3 type of blood vessels. Rather than not only it containing the artery, we call as hepatic artery. This is not the artery. You want to mention hepatic artery. Hepatic artery. And then another one is hepatic vein. And then another one is portal vein. This is a new that you will never see other parts of the organ one. This is the only special blood vessel that found in the liver only. So what are their function? So hepatic artery and the hepatic vein actually are totally same. What are the same as the other organ? Hepatic artery is to carry oxygenated blood. But and the hepatic vein is carry deoxygenated blood. So your oxygen gas will go at here and then will release out, right? And then all the cells. So this is all the cell. The liver cell will carry out metabolic activity, right? So they will release all the carbon dioxide and metabolic waste to the vein, and then this will carry to the lung, back to the lung. But how about this portal vein? So this portal vein is very special. If you still remember that you already learned in SPM about the assimilation of your liver, you know that in your small intestine, absorption of the nutrient in small intestine contain 2 parts, right? Either it absorb the water dissolved material or absorb the fatty water dissolved nutrient or fatty dissolved nutrient. Water dissolved nutrient will go to the blood vessel, and fatty dissolved will go to the lymphatic vessel. This one you already learned. I just recap to you guys in SPM. So bunch of portal vein is the blood vessel coming from the small intestine, and then this one is carrying out all the nutrient that absorbed in the intestine already. We know that the main function of your small intestine, duodenum, small intestine is to absorb nutrient, right? And then all the water dissolved organic nutrient or any nutrient will be absorbed into the blood vessel. So this one is portal vein. So all the nutrient will be transported through the blood vessel from the small intestine to the this portal vein. So this portal vein is the blood vessel that carry nutrient from the small intestine. So this is another type, another blood vessel lah, I can say. Okay, so these 3 you need to know. So about the location, so when in examination come out this kind of diagram, you need to label. So hepatic portal vein and hepatic artery are located near to each other one. Whatever the size may be different. So maybe in examination dia akan keluar size macam ni lah. Maybe dia akan hanya keluar circle size je. So you need to know that portal vein and artery are located each other. So if you see there are 2 circle at there, so memang ini adalah portal vein. Why? One of them is artery, one of them is portal vein. Why? Because you see that after they go to this junction we known as sinusoid, and also this junction we know as sinusoid. Yeah, ini. You see, this junction is recalled as sinusoid. When they already enter the sinusoid, the blood are mixed together. The blood from the hepatic artery and your portal vein are mixed together, and then it will go to the central vein. So this one is recalled as central vein lah. So in the structure, this basic unit of structure, you will add on the branch because this is a branch of the portal vein and also branch of hepatic artery vein. And then this one is branch of hepatic vein or we know essential vein. So this is the general structure, and also I need to tell you is this cell. You see that there are many color cells, right? So this is the cell we call it, this is the liver cell. So in examination you cannot write liver cell. There are the scientific terms you need to learn. Hepatocyte. Hepatocyte is the scientific terms of the liver cell. So you cannot mention liver cell, you need to mention hepatocyte. Okay, so this is a basic unit of the liver structure, so you need to know because maybe structure question coming out. But actually not only that. Okay, so this is all the explanation. Maybe when it coming out in essay, then you need to mention like this you need to all like that So but I think MPN tak akan boring like that coming out want you to describe the structure of liver, but it is also possible to come out. When it coming out, you need to explain all this So I already mentioned about the hepatic artery, hepatic portal vein, and also hepatic vein, right? Okay, so and then hepatocyte, this is the liver cell, and then this hepatocyte is the place site of the biochemical reaction. Later you will learn about the function of your liver, about the carbohydrate metabolism, and also protein metabolism inside. All this is happening inside the hepatocyte. So normally this hepatocyte containing nucleus and rich in mitochondria, Golgi apparatus, and also lysosome, especially mitochondria. Okay, and then the hepatocyte are tightly packed to each other, and their surface are contact with blood vessels, have microvillus, are used for material exchange. So this structure does not show microvillus structure, but actually if you see under microscope, will show the microvillus structure this in exam normally it will coming out like this it will not coming out like that, but normally in exam they are coming keluar macam ni, but actually similar je. You see these two kinds. So the lower part is outer vein, the upper part is portal vein. So kenapa? Kerana ini awak boleh tengok dua-dua ni kan, lepas dia gabung together. So kalau tengok macam ni, then totally yang upper portal vein, lower hepatic artery, and then yang tengah, yang ini the opposite, another opposite side is branch of hepatic vein. Okay, and then you see these all these, the flow of these we call as interlobular vessel. Lobular vessel. So this this line, when the blood vessel of the hepatic portal vein and your hepatic artery are mixed together, this one we call as interlobular vessel. And then when they reach the gap, when there are the junction at here, we will known as sinusoid. Sinusoid is the junction, you can say it is a pass through the sinusoid which drain the blood between the cord of the hepatocyte. So like a junction, when they near the junction we call as sinusoid. So normally your material are exchanged at there also. And then your intralobular vein or your central vein located in the central of the lobule receive blood from the sinusoid and pass into the hepatic vein. And then arrangement of your blood vessel, so this is a basic unit, functional unit we call as sinus. So when your all the hepatocyte or blood vessel, artery and your portal vein, hepatocyte and your sinusoid are combined to each other, we known as the basic unit we own as liver as sinus. This is a functional unit of the liver. So another one of the function of the hepatocyte is also will produce bile. It will also secrete bile. So this bile will store at the gallbladder. So we know that bile, the function of bile, function of bile is to like an enzyme, it will increase the digestion rate in your small intestine, right? So this one you already learned in SPM, so I don't want to repeat again and again. So this so you will see that rather than when you go to blood vessel from the portal vein and also hepatic artery, you will see there are also another vessel at here. So this one is not a blood vessel. You need to know this one is not a blood vessel. This one is we call as bowel canalicus. Because your hepatocyte will secrete the bowel, your bowel, and then it will go, it's secret, and then it will go through this bowel canalicus, and then this one is one type of vessel, and then this bowel will flow into the bowel duct. Went to the bowel duct, and then this bowel will store at the gallbladder. Okay. Another one, there are also another structure we call as Kupffer cell. This one, it is located at the hepatocyte, like that. Kalau dia ada keluar macam ni, ada hitam-hitam, kalau macam hitam-hitam macam ni, or in general structure, you see in hepatocyte there are the spatial structure found on the surface of your hepatocyte. This one we call as Kupffer cell. So what are the function? This function is normally to eliminate bacteria and also old erythrocyte by phagocytosis. So this is the main function. Okay, so that's all about the liver structure. Okay, so about the liver structure. Okay, next I go to the most important part, where the essay question always coming out, liver function. So in liver function, there are two main functions that you need to learn: carbohydrate metabolism and also protein metabolism. You feel familiar with it, right? Chapter in semester 1, chapter 5, you learn about carbohydrate metabolism, glycolysis process, and also you learn about alternative of product, protein metabolism. So in this topic, you will learn again about this, but you don't need to memorize all the Krebs cycle etc. No, but there are another mechanism that you need to know. So in carbohydrate metabolism, there are three mechanisms: glycogenesis, glycogenolysis, and also gluconeogenesis. So in semester 1, chapter 5, you learn about glycolysis. It is a breakdown, oxidation of the glucose molecule. Glycogenesis, so what is the meaning? Glycogenesis, meaning that it is formation of your glycogen. And then glycogenolysis, you will see the lysis, you refer to glycolysis, right? You will still remember glycolysis. Lysis is the breakdown, right? So glycolysis is breakdown of your glucose molecule. So glycogenolysis is the breakdown of your glycogen. And then gluconeogenesis, so this one, genesis, is a formation process, meaning it want to form back the glucose molecule, formation of the glucose molecule. And then also protein metabolism, deamination process, ornithine cycle, and also transamination process. So there are the different mechanisms you need to memorize as well, but for me it's more easy compared to chapter 5 lah. Okay, so carbohydrate metabolism. So in this one is that you already learned in SPM. You know that if you want to about the negative feedback mechanism of your blood glucose, you have two hormones, right? Either will be secret by pancreas. So there are the spatial cell we call as beta cell of islet of Langerhans. So this one you memang awak kena hafal. Not in your SPM level you just say pancreas, right? But in STPM you need to mention beta cell of islet of Langerhans because got marks. This got marks one. If you say pancreas only, no marks will be given. Ini ada satu markah tau. Awak mention ni ada satu markah. Kalau awak mention pancreas then no marks lah. Okay. So insulin is play the role to decrease your blood glucose level. When your blood glucose is very high. So this hormone will trigger the mechanism we call as glycogenesis. Glycogenesis meaning you need to form the glycogen. You know that insulin in your SPM level, you just say that glucose will be converted to glycogen, right? You know that in polysaccharide structure that you already learned in semester 1, you know that polysaccharide we have 3 polysaccharide, right? Starch, glycogen, and also cellulose. And then starch is the carbohydrate, the polysaccharide that's stored in the plant. And then in human, in animal, the polysaccharide that be stored is in form of glycogen, and then glycogen is formed from the multiple unit of glucose molecule that you already learned in semester 1. So glycogenesis is the process how all the glucose molecule condense together to form the glycogen molecule. So this is the whole role of insulin. And another one is glucagon. So glucagon is the hormone when your blood glucose level is very low. So meaning that you are in very hungry state, or you are in fasting state. Like puasa lah. You doing puasa, right? Doing puasa, Ramadan puasa, awak tak ada makan dari pukul 5 ke 6 ke 6 sampai malam 7 tak makan, malam 7 baru boleh makan kan. So at this kind of period, glucagon will be secreted out, because at this kind of period your blood glucose level is very low. So you need to maintain your blood glucose level. You need to your pancreas, which is another cell we call as alpha cell of islet of Langerhans, will secrete the glucagon. So again, you cannot just mention in pancreas only, you need to mention alpha cell, because got 1 marks as here. If you just mention pancreas, so no marks you can mention but no marks okay. Glucagon is alpha cell. So this alpha cell will secrete glucagon, and then this glucagon will be transported through the blood, and then will go to the liver. Both hormones will go to the liver. Both hormones are secreted from pancreas, and then this hormone will be transported through the blood. You still remember the last chapter, chapter 9, hormone topic. But these two hormones both are non-steroid hormone, right? Non-steroid hormone mechanism. Kena relate lah, kena relate. Semua kena relate. Both are the non-steroid hormone. Both are the non-steroid hormone, right? Hello. Okay. I think my mic is have. Hello, testing. Why my mic? Hello. Hello, testing. Hello, test. Testing. Okay. Okay. Just now I say about these two hormones are the non-steroid hormone, right? Because these two hormones are the peptide hormone. They are protein hormone. So these two hormones also will undergo non-steroid mechanism. So also there are possibility in exam they will come out these two type of example of hormone. Want you to write out the mechanism of the insulin. Mechanism hormone in insulin. So you need to know lah. Insulin is non-steroid hormone and similar as the glucagon lah. Okay. So you need to all related. So glucagon is secreted at alpha cell, and then it only will be stimulated, will be secreted when your blood glucose level is very low, because it need to increase your blood glucose level. And then in SPM level, you know that glucagon is to convert glycogen into glucose molecule, right? This is SPM level that you know. So similar, the role is same, but there are two main mechanisms that you need to know are glycogenolysis and also gluconeogenesis. These two mechanisms play the role to convert glycogen to glucose molecule, but the conversion of glycogen to glucose is glycogenolysis. Gluconeogenesis are not conversion of the glycogen to glucose. It is the conversion of your pyruvate to glucose molecule. Later I will explain it. So But you need to remember that in glucagon hormone, it will trigger these two. But insulin only will trigger glycogenesis only. But glucagon will trigger glycogenolysis and also gluconeogenesis. Okay. Okay, so this diagram, I think everyone know lah. I just repeat lah. I think I already explained all lah. This is a negative feedback mechanism that you already see in SPM level 2, right? Blood glucose level, normal blood glucose level. And so when your blood cell is when your blood glucose level increase, your beta cell of your islet of Langerhans in pancreas will secrete insulin, and then so that secrete insulin. So in secret insulin, so your liver was starting to store convert the glucose and store the carbohydrate in terms of glycogen lah, right? And then after that, your blood glucose level will increase your set point, and then to decrease the increase the insulin level lah, and also this one glucagon. Okay, so this is SPM. So in STPM, awak tak boleh sentuh macam ni je, cannot, because you need to explain the mechanism of your glycogenesis. You need to explain it, because in syllabus, you will refer to syllabus, they are already mentioned, describe glycogenesis, glycogenolysis, and also gluconeogenesis. So all these mechanisms you should write in the essay. Okay, so glycogenesis, I already mentioned, is the process when the insulin is secreted. When the insulin is coming to the liver, so it will induce this mechanism, glycogenesis. So I already mentioned, it is a formation of glycogen from 1,000 molecule of glucose molecule. So you will still already forget apa itu glycogen. Inilah glycogen structure. I just recap the, but I think inilah glycogen. So we know that glycogen are the polysaccharide which is highly bond structure, right? Highly bond structure, and there are two bonds, right? Alpha 14 glycosidic bond and also alpha 16 glycosidic bond. Due to the pressure of alpha 16 glycosidic bond, cause the glycogen become highly bunched and highly compact structure. So glycogenesis is the process how all these single unit of monomer glucose molecule condense together to form glycogen. And maybe you will ask, condensation process lah, right? Okay, all condensation process, but how to form alpha 16 glycosidic bond? You will still ingat tak? You never learn yet. Glycogenesis you will learn. Okay. Okay. So there are many steps. All these steps you need to know. Okay. Where are? Okay. So the first step, of course, glucose lah. Memang glucose lah kan, insulin high glucose level. So you need to change your glucose. You need to condense your glucose into the glycogen. So first of all, occur glucose. So your glucose molecule will go to the will be phosphorylated to become glucose 6-phosphate. Glucose 6-phosphate. So this is the first process of glycolysis, right? You still remember, right? So meaning that there are phosphate molecule phosphorylation process from the carbon number 6 of your glucose. So the enzyme using is hexokinase. Okay, so this is the first process. After that, glucose 6-phosphate will be isomerized to become glucose 1-phosphate, meaning that the phosphate group will be changed to the from the carbon number 6 to the carbon number 1. Just change only lah. So glucose 6-phosphate will be isomerized to the glucose 1-phosphate. Okay, starting step 3 is more complicated because it will involve another molecule we call as UTP, which is a uridine triphosphate. UTP. This is a totally new molecule, another molecule. Why we want to use this molecule? Because it is act as the energy activator to activate glucose 1-phosphate first before the synthesis of the glycogen. Because you want to form the glycogen, it is just like not like the formation of maltose molecule, simple combine. You don't imagine that it is like the simple process like the formation of maltose molecule, just combine banyak maltose sampai dia jadi glycogen. No, there is no such thing like that. No, simple like that. It is very complicated process actually. So this is another molecule we call as UTP molecule. Okay, so this UTP molecule will react with the glucose 1-phosphate to form UDP glucose. This one. So meaning that it will breaking out 2-phosphate group within UTP by UDP glucose phosphorylase. So meaning that glucose 1-phosphate will react with UTP, and then this UTP contain 3-phosphate group, right? So why we call as UTP karena triphosphate, ya, ada 3 phosphate. Sekarang because reaction occur, this 2-phosphate group in the UTP will be released out. Dia akan keluar. Why like that? Okay, all these UTP, the 2-phosphate group will be released out, and then will bind together. So the phosphate group of the glucose 1-phosphate will be bind to the UDP. So it will form the UDPG. The full name is uridine diphosphate glucose. UDP is uridine triphosphate, right? So now because it released 2-phosphate group, so now it only remaining 2-phosphate group. One come from UDP itself and one come from glucose 1-phosphate itself. So they are containing total 2 phosphate group and scale UDPG. Okay, next, what are the next step? Okay. So this step is more complicated again. Now is more complicated. Okay, now you already have UDPG at here, right? UDPG. This is a UDPG structure. UDP group. This is glucose, right? And then this is UDP uridine structure. So your addition of your UDPG to a growing chain of glycogen is the next step in glycogen synthesized by using glucose glycogen synthase to form 1,4-glycosidic bond. So actually, you in general, you can directly use glycogen synthase to form 1,4-glycosidic bond already. But more complicated. Enzyme ni memang geram lah, tak boleh. Dia cakap tak boleh. Enzyme ni cannot simply form a bond between these 2 isolated glucose molecule. It only can add into existing chain that have 1,4-glycosidic bond. So meaning that in the general, these 2 molecule UDP glucose, if you want to form 1,4-glycosidic bond to another UDP glucose, cannot, tak boleh punya. Kerana enzyme ni glucose synthase tak boleh. Dia tak boleh function macam ni. Dia enzyme ni hanya boleh form dia ada glycogen. Dah ada glycogen punya 1,4-glycosidic bond. Maksudnya macam ni lah. You still remember yang ni. Kalau dia glycogen synthase ni dia hanya boleh add UDP glucose dekat sini je. Kerana dia yang ni dah ada 1,4-glycosidic bond. Hanya glycogen synthase hanya boleh add an addition UDPG dari sini. Kalau ada dua UDPG yang tak ada 1,4-glycosidic bond, enzyme ni tak boleh. So how to resolve this problem? Okay, so our body is very smart. The glycosynthase tak boleh macam ni, so dia akan carilah alternative way. So the alternative way is the initiation of glycogen synthesis will require primer known as glycogenate. Inilah we call as glycogenate. This is a primer. Dia kena ada primer, satu support lah. You can say that ni macam satu support. Kerana glycogen synthase dia tak boleh. Dia tak boleh directly form the bond, form the 1,4-glycosidic bond between these 2 isolated UDP glucose. Tak boleh. So require primer. So the primer we know as glycogen. Inilah glycogen structure. Okay, so glucose residue first will link to this tyrosine hydroxyl, and glucose residue are successfully added to this first one. So you see this diagram, this is a UDPG, right? UDPG. So when the glycogen is added to here, it will release UDP molecule, and then at the same time, another UDP glucose molecule will be added. So it will form this structure. So ini first glucose molecule, first UDP glucose, they bind the glycogen primer, and then it will glycogen, and then it will release this glucose molecule will release the UDP. Ini yang dia akan release, and then at the same time another UDP glucose will bind to it to form the 1,4 glycosidic bond at here. But this UDP glucose will not release the UDP. And then the first UDP glucose molecule, still bind with the glycogen. So there are another enzyme we call as glucosyltransferase enzyme. So this is the enzyme that play the responsible to formation of the 1,4 glycosidic bond. And then this glycogen molecule will act as the catalyst for the addition of glucose. So this process will be repeated until there are 8 molecules. 8 molecules like this. When there are 8 molecules, 1, 2, 3, 4, 5, 6, 7, 8 until here, when the addition of the UDPG until 8 until here, then your glycogen will be discard, dia akan keluar, then the glycogen synthase will be take part this problem. Will be replace the function of the glycogen. So it will can directly add the UDPG to the remaining glucose molecule lah. So awak kena tahu lah. So hopefully you guys understand it. Okay. So once they already form 8 molecules. So now we know that glycogen is bunch. So it is not the single stranded only, no single bunch only. So there are the bunch too. So how to form the bunch? So there are the bunching enzyme. The bunching enzyme will break down the 1,4 glycosidic bond at the bunch point and transfer these oligosaccharide fragment into another chain by catalyzing the formation of 1,6 glycosidic bond. So you will see this diagram, ini dah form satu bunch lah. So kalau dia add glucose sampai sekarang, okay, their bunching enzyme will detect the bunch point. When it detect the bunch point, it will cut down this 1,4 glycosidic bond, and then this enzyme akan bawa semua ni oligosaccharide kepada yang ni lah. Another chain will transfer to another chain, and then to form the 1,6 glycosidic bond. So this one is the formation of bunching. So until here, glycogenesis is end. So glycogen is formed now. Okay. So this is the overall process of your glycogenesis. Okay, so any questions so far? Clear, right? Okay. So I think everyone clear because no question. Okay. So everyone clear. So I will proceed to glycogenolysis. Okay. We already talked about the glycogenesis, which is the formation of the glycogen. This is a place when the insulin is secreted and trigger the mechanism, because you need to increase the blood glucose level. So you need to condense all the glucose molecule into the glycogen and store in the liver. Glycogen will be stored in the liver. Okay, when the blood glucose level in your blood is very low, and then normally in fasting state, so glucagon will be secreted from the alpha cell, and then this glucagon will transported into the liver, and then it will trigger the two mechanism by glycogenolysis and also gluconeogenesis. So one of the first step is the glycogenolysis. So it is a reversible reaction about the glycogen. So it is a breakdown of glycogen because you need to increase your blood glucose level. So you need to large amount, you need to increase your glucose concentration in your blood lah. So this is the overall process about the glucogenolysis. So for this process, it is more simple compared to the glucogenesis. Glucogenolysis are more simple because it is the hydrolysis process, right? Okay, so this is the first step, glycogen. So if you see here, glycogen. So glycogen, you know that it is linked with 1,4-glycosidiol can cleave with the glycogen phosphorylase through the phosphorylases process to glucose 1-phosphate. So you will see glucose 1-phosphate. So this is the directly, meaning that all the glycogen yang ada glycogen dekat sini, it will using the enzyme we call as glycogen phosphorylase. So the glycogen phosphorylase will break down this 1,4-glycosidiol to form glucose 1-phosphate. So after that, this glucose 1-phosphate will go to reversible reaction lah. Tadi is glucose 6-phosphate to glucose 1-phosphate lah. So sekarang dia tukar balik isomerize back to glucose 6-phosphate, and then it will go back to glucose molecule. So this is the enzyme. These are the enzyme. So glucose 1-phosphate will isomerize back to glucose 6-phosphate using the glucosamine, and then glucose 6-phosphate will be phosphorylated to form glucose back to using the glucose 6-phosphatase. Okay, but we know that glycogen containing branching enzyme. So all this branch, if there are many branch, so the remaining branch, they will break down by the glycogen phosphorylase. But when there are remaining four glucose, limit branch at here, there were another enzyme we call a transferase. It will transfer all these remaining three glucose molecule at here, transfer back to the another change. So you see this explanation. So glycogen phosphorylase only will break down 1,4 until it will stop the 4 residual glucose from the branch point, which is the limit branch. So this is we call as limit branch. So hence your transferase will break the 1,4-glycosidiol bond on the limit branch that have only one residue that link with 1,6-glycosidiol. So it will break down this and then transfer these. The debranching enzyme will cleave the 1,4-glycosidiol to release free glucose. So this I just want to say this transferase will transfer this enzyme to the another chain this, and then there are another enzyme we call as the debranching enzyme. So normally it's alpha-1,6-glycosidase. It will break down this 1,6-glycosidiol and then go to forming the glucose 1-phosphate . So this glucose 1-phosphate will later be isomerized back to glucose 6-phosphate, and then the remaining branching, it will using the glycogen phosphorylase again, to form glucose 1-phosphate and then isomerize back to glucose 6-phosphate. So more easy, just that the hydrolysis process not only using one enzyme because it known as glycogen phosphorylase. Just that the branching, this enzyme will only hydrolyze until it will remaining the 4 residue near the limit branch point. Near the branch point, so that it will form the limit branch, and then there are another enzyme called a transferase. It will transfer these 3 limit branch to the another change, and then this another one residue will be break down by the debranching enzyme, and then it will form glucose 1-phosphate and then repeat again. So that's all. Glycogenolysis. Easy right? Sangat senang je lah. Okay. So, any questions so far? Okay, no question. Okay, now we will proceed to gluconeogenesis. Okay, so sorry. Okay, gluconeogenesis. So it is the formation of glucose from pyruvate or any non-carbohydrate resources. So this happening when the glycogen is used up. When there are no glycogen inside your liver already. Because the first trigger mechanism from the glucagon is trigger the glycogenolysis first. But when your glycogenolysis is already used up, finished up, meaning that glycogen is already totally used up, there are no glycogen inside your liver already. So gluconeogenesis will be take place. So this process, what is this? So it is the formation of glucose from non-carbohydrate resources. So normally it's pyruvate lah. Well, you ingat balik similar ke? Ingat tak? Glucose to pyruvate glycolysis, right? So it is a irreversible reaction, reversible reaction of the glycolysis. But ada sedikit perubahan. So although you already enter semester 2, but glycolysis process you need to still memorize it. Kerana gluconeogenesis is the reversible reaction of the glycolysis. However, there are some differences compared to glycolysis. There are the first step, second step, 9th step, and 11th step are different. Because if you still remember, this is the glycolysis process, right? You still remember? I think you already forget. Never mind. You see this again, you remember it. You still remember that in the first step of your glycolysis, glucose to glucose phosphate, this is the irreversible reaction, the first step, and then the third step, fructose 6 phosphate to fructose 1,6 phosphate. This is also irreversible reaction. And then a final step, phosphoenolpyruvate to pyruvate. Pyruvate also. Sorry. Why like that? Okay, you still remember that first step of the glycolysis, glycolysis, first step, third step, and your final step, I think is 10, right? 10 step are the irreversible reaction, meaning that it is irreversible. You cannot terbalik punya. Dah boleh terbalik. Yang lain remaining step are reversible reaction. You can use same enzyme. But these three process, the first one, the second, the first, the third, and the final, the 10th step are all the irreversible reaction. So if you want to reverse back, how? So you want to reverse, you want to convert pyruvate back to phosphoenolpyruvate. Macam mana? Tak tahu. So our human body are very smart. So they have their own process. So this yellow color highlight one I already enlarged. So these are the alternative way to convert back to ensure that they have a reversible reaction. Okay, so these reactions are totally different as well, and the enzyme used are totally different. Okay, so the first step, you will still remember the glycolysis. The final step of glycolysis is convert phosphoenolpyruvate to pyruvate. Right. Okay, but in the first step of the gluconeogenesis, you cannot directly convert pyruvate to phosphoenolpyruvate. You need to have an intermediate step. So there are 2 steps. You need to convert pyruvate to phosphoenolpyruvate. They require 2 steps. And then this process is only occur in mitochondrial matrix. You will see the key. because you see this pyruvate will convert to become oxaloacetate, right? And oxaloacetate is the intermediate in the Krebs cycle. So Krebs cycle occur at where? Krebs cycle occur at mitochondrial matrix, right? So this process is only can occur in mitochondrial matrix only. Because oxaloacetate is only found in mitochondrial matrix. It cannot diffuse into the cytoplasm one. Okay. Now the first step is your pyruvate will use ATP using the pyruvate carboxylate enzyme undergo carboxylation process to form oxaloacetate. Okay, so the key point is it will use pyruvate carboxylase for using ATP and then form oxaloacetate, and then this process is occur at mitochondrial matrix. After that, oxaloacetate will use GTP again, using the GTP to form back phosphoenolpyruvate. So it requires intermediate step. It cannot directly, you want to convert pyruvate to become phosphoenolpyruvate. Cannot. Gluconeogenesis cannot. You cannot. You only can. You need to have an intermediate process like organic chemistry lah. Play the in terms of their structure. So you need to form oxaloacetate first, and then using the GTP inside the mitochondrial matrix, form back phosphoenolpyruvate. So this process is decarboxylation process. Release one oxygen lah. Okay. One carbon dioxide. Sorry. Okay. So this phosphoenolpyruvate later will diffuse out from mitochondria to cytoplasm. So starting step 3, starting here, phosphoenolpyruvate sub 3 until here, semua ni lah phosphoenolpyruvate, 3 phosphoglycerate, 2 phosphoglycerate, 3 phosphoglycerate, 1 3 biphosphoglycerate, and then glycerate aldehyde 3 phosphate. All these processes are reversible. So all the enzyme, just that all are reversible lah. So you see this, if glycolysis it will produce ATP, right? So in reversible process it will use ATP lor. So all the enzyme, all the process are similar. So this one will occur at cytoplasm. Starting from step 3, when the phosphoenolpyruvate is decarboxylated from the oxaloacetate, it will diffuse from mitochondrial matrix to the cytoplasm, and then starting from here, it will start to reversible reaction. Go through the reversible reaction until it go to the step 9, which is the third step of the glycolysis, where in glycolysis, where glucose phosphate need to convert to glucose phosphate need to phosphorylated to become fructose 1,6-phosphate, right? But in glycolysis, this process is irreversible. So it require another enzyme we call as fructose 1,6-biphosphatase. Another enzyme, glycolysis enzyme, saya dah lupa lah. I already forget. But this is the new enzyme it require for the phosphorylation process lah. About the step 3 of glycolysis, but step 9 of gluconeogenesis. And then another one is final step, step 11, where glucose 6-phosphate convert to glucose. So it require glucose 6-phosphatase. So this one is similar to the glycogenolysis process, right? Glycogenolysis, you also convert glucose 1-phosphate, and then you convert isomerize back to glucose 6-phosphate. So this glucose 6-phosphate will use the glucose 6-phosphatase through the dephosphorylation from back glucose molecule. So this is overall about the gluconeogenesis. But in examination, you not just mention these 3 steps. You should mention all this. You need to mention all some of the reversible reaction of the glycolysis. Okay, but the most important thing is first step, second step, 9th step, and also 11th step. Okay, so that's all for the gluconeogenesis process. Okay, next is about Krebs cycle. Okay, so this is the another process. Normally is when the people doing exercise. when you are doing the exercise, if you still remember in your chapter 1 to anaerobic respiration, you know that you are doing the exercise, you will undergo your muscle cell will undergo anaerobic respiration and then produce lactic acid, right? Anaerobic respiration. So you know that how this lactic acid can eliminated. Macam mana dia eliminate? Dia tak akan stay at your muscle tissue, and then dia tak akan stay at your muscle tissue and stay at liver one. Dia kena eliminate because this is one of the I cannot say toxin, but this is a useless product lah. It is one of the useless product to our human. We not use it. So memang either eliminate, you kena eliminate. So macam mana eliminate? So we eliminate through this Corwick cycle. Okay, so this cycle is how this happen. So you still remember in muscle tissue, in the anaerobic respiration, lactate fermentation, you know that glucose will go through glycogenesis, go form pyruvate, and then form lactic acid, right? And then this process you already learned in semester 1. I don't want to repeat again. You can refer back, and in examination you need to mention it. Okay, mention it semua okay. After that you form lactic acid. Okay, so this lactic acid later it will be diffused out from muscle tissue to the blood vessel and then go transported into the liver. It will transport to the liver. Okay, so this liver lactic acid will convert back into the pyruvate. So it will reversible reaction lah. So in lactate fermentation you use NADH. So now you will produce NADH. Right, and then this pyruvate will go through gluconeogenesis to form glucose. So this is what we call as Corwick cycle. So this one is also one of the mechanism function occur in the liver. So this one is normally occur when you are doing exercise. When you are doing exercise, when you have the high amount of lactic acid, and then this glucose molecule formed in the liver later will be transported to the muscle tissue. Because muscle now is require high amount of glucose, because muscle now is highly reactive. You need to do you are doing exercise, right? So your metabolic activity or your muscle is very high. So your muscle is require a higher amount of glucose. So it will take the glucose from liver. Okay, so this is the simple process of Corwick cycle. Okay, next I will proceed to protein metabolism. Okay, so in protein metabolism, I think in semester 1, chapter 5, about the alternative energy, I think you already mentioned sikit-sikit lah. Awak dah tahu. I assume you already know about the protein metabolism alternative energy. You already learned in semester 1. You will know that you have deamination process, right? And then you will go through ornithine cycle. And there are also another step we call as transamination that you already, but you don't know the process. You just know the name in semester 1. Okay, so in this topic you will learn about more detail, and this process is only occur in liver. Only occur in liver only. So that is why that protein metabolism is only occur in liver cell only. Others part tak boleh. tak boleh. No, other cell cannot metabolize a protein. They only can use amino acid. But if you want to metabolize protein, the huge protein molecule become amino acid, only the liver cell can do it. Okay, so what are the process? So the first step is about the deamination process. So deamination process is you need to remove amino group from amino acid to form ammonia, which is very toxic and soluble. We know that. Also in semester 1, we know that the basic structure of amino acid we containing amino group and also carboxyl group, right? And then amino group is actually the basic structure is ammonia lah, and we know that ammonia is very toxic. It is very toxic to human, useless one. So you need to remove this first. So deamination is to remove amino acid to form ammonia. Okay, so you see this diagram lah. Diagram. Okay. So either you form ammonia or ammonium ion, either one. Both are acceptable. So I use glutamate as example. So this is a glutamate amino acid, glutamate, and then glutamate. So it will undergo deamination process, process to release out the ammonium ion. The most important I need to highlight is ammonium ion. So this ammonium ion will be degraded, excreted out through the urine. So it will go to the kidney. Later I will explain it tomorrow. Osmoregulation topic. This ammonium ion will be transported to the kidney and excreted out as urine because this is a very toxic particle. We don't want it. The human don't want it. It is very toxic. Okay. And then after that, how the ammonium ion be secreted out through the urine? This is a process we call as ornithine cycle. Because ammonium ion dia tak boleh be secreted out one, tak boleh. Dia kena form urea. Only urea. Dia kena form urea. Urea semua orang tahu kan? Urea is one of it, also is the toxin, and it is coming from the protein which or ammonia. So once they form the urea, and then this urea will be excreted out in terms of urine . Okay, so this ammonia formed from the deamination process will be added with the carbon dioxide first. So yeah, so this is the ornithine cycle first step. This is the condensation process. So meaning that your carbon dioxide and your ammonium ion or ammonia, both are suitable, will be added to each other to form carbamonic phosphate. This is the first step, and this process is occur at the mitochondria, liver mitochondria. This process first step carbamonic phosphate is occur at the mitochondria. Okay. And then after that this carbamonic phosphate will be add with ornithine. This one is ornithine, right? Carbamonic phosphate will be add with ornithine. Ornithine to form citrulline. Okay, repeat again. Carbamonic phosphate you already formed in step 1. It will be add with ornithine to form citrulline, and this process also occur in mitochondria of the liver. Liver mitochondria, bukan mitochondria yang normal punya. Liver, you need to mention liver mitochondria. Okay. And then after that, this citrulline will be diffused out from mitochondria. So afterward, this process is occur at the cytoplasm of liver cell. So repeat again. First step and second step are occur in mitochondria, and then after that, this process, the citrulline will diffuse out from mitochondria to cytoplasm of the liver. And then the third step okay. The third step is it should be ini typo. I don't know why I copy many. Okay, but the third step, this is the third step. Third step is the citrulline. Once your citrulline is already go to the cytoplasm, it will be add with aspartate. Citrulline will be add with aspartate and using one ATP molecule to form argininosuccinate. Repeat again. The third step, once the citrulline is already transported into the cytoplasm, it will be add with aspartate to using one ATP molecule to form argininosuccinate. Okay, this argininosuccinate later it will undergoes hydrolysis process. Not hydrolysis. Lysis process. We will break down to form fumarate and also arginine. Break down become one side product is fumarate and also arginine. And then this fumarate will go to enter Krebs cycle. This is an intermediate of Krebs cycle, right? So it will go to Krebs cycle lah. Fumarate will enter Krebs cycle. But this arginine will continue ornithine cycle. Okay, so this arginine, okay, later this arginine will be hydrolyzed using water, hydrolyzed to form urea as a side product, and then form back again ornithine. And then this ornithine later it will transport back to the mitochondria too. So urea is the final product. So this urea later will be excreted out through the urine, and it will be transported to the kidney and excreted out in terms of urine. So it requires water molecule, hydrolyze lah. Okay. So that's all for the ornithine cycle. So repeat again lah. So I use this diagram lah. First step is the deamination process, where the amino acid you need to remove all the amino groups, so you will form ammonia and ammonia ion. And then this process ammonia will be added with water or carbon dioxide using 2 ATP molecule to form carbamoyl phosphate. And then this carbamoyl phosphate will be added with ornithine to form citrulline. And then this process is occur at mitochondria of liver. And then this citrulline later will diffuse out from the mitochondria into cytoplasm. So once it already enters cytoplasm, it will be added with aspartate using 1 ATP molecule to form argininosuccinate. Okay, then this argininosuccinate will be lysed to form 2 products. One is fumarate and one is arginine. And then fumarate will be enter Krebs cycle, and arginine will continue the cycle. So this arginine later will be hydrolyzed using water molecule to form urea and form back again ornithine. And then this urea is the toxic product that we don't want, and then it will transported into the blood, transported through the blood to the kidney, and then will be excreted out in the form of urine. And then this ornithine later it will diffuse back into the mitochondria, and we add again the carbohydrate phosphate to proceed another turn of ornithine cycle. Okay, so